Hello, everyone, and welcome. My name is Rula Deeb. I'm a senior principal at Geosyntec in Oakland, California, and the coordinator of the webinar series on behalf of SERDP and ESTCP. I have the pleasure of facilitating today's event. This webinar focuses on DoD-funded research efforts to develop innovative tools for PFAS sampling and monitoring. First, Dr. Rainer Lohmann from the University of Rhode Island will discuss his work on developing and testing PFAS passive samplers in groundwater, surface water, and pore water. Second, Dr. Craig Devine from Arcadis will talk about the assessment of a novel multi-port well system to help in PFAS site characterization efforts. Both presentations will be followed by a brief Q&A session, and we will conclude the webinar with a longer Q&A session. The next several slides provide instructions on optimizing your webinar experience. If you have not done so already, please download Zoom at the link shown here and also provided to you in your webinar registration confirmation email. If you're not able to download Zoom, you can view the slides using a compatible internet browser such as Firefox, IE, or Edge, and by creating a free Zoom account. If you continue to have difficulties or if your screen freezes, please try keying in Control and F5 to perform a hard refresh of your screen. If you're accessing the audio through your computer, click the arrow next to the Join Audio button, select Test Speaker and Microphone, and follow the prompts as they appear on your screen. If you continue to have difficulties, just call into the conference line shown here or submit a comment using the chat box. Only use the chat box for comments related to technical difficulties. The Q&A option should be reserved for questions for our speakers. And in case of continued difficulties, you can always download a PDF of the slides from the webinar webpage and call into the conference line in your webinar registration confirmation email. Keep in mind that we will be live streaming the webinar, or we are live streaming the webinar on the SERDP ESTCP YouTube channel at the link shown here. Just a few reminders, today's broadcast will be listen only. Please submit questions using the Q&A box on your screen. You do not need to wait until the Q&A period to submit your questions. We do encourage you to get them in well in advance of the Q&A sessions. And when you submit your questions, please add your organization name at the end so that we can identify it. With that, I would like to introduce Dr. Andrea Leeson, who is the Deputy Director of SERDP and ESTCP, as well as the Program Manager for the Environmental Restoration Program area. Dr. Leeson has been with SERDP and ESTCP since 2001, and prior to that, she served as a research leader at Battelle, where she conducted scientific research on in situ bioremediation and the design and implementation of innovative biological, chemical, and physical treatment technologies for both site remediation and industrial wastewater. Andrea received her doctoral degree in environmental engineering from the Johns Hopkins University. Andrea, please proceed.
Hey, thanks so much, Rula, and thank you to everyone who's able to join the webinar today. I'm very quickly going to turn you over to our technical presentations today because I know that's what you tuned in for. But before we do that, I would like to give you a little bit of an overview about who we are in terms of our two programs that have supported the work that you'll be seeing today. We have two companion programs. The first is SERDP, the Strategic Environmental Research and Development Program. This is a traditional R&D program. It is a partnership between the Department of Defense, Department of Energy, as well as the Environmental Protection Agency. And very specifically, what we're doing is to develop technologies to address the near-term needs in the realm of environmental management. The companion program is ESTCP, the Environmental Security Technology Certification Program. And this is a demonstration validation program, which is a little unique to most funding agencies. And here we take technologies or knowledge that have matured out of SERDP or other R&D programs and we look to transition that technology out of the lab into the field and establish the cost and performance for these technologies. Ultimately, we want to accelerate commercialization of the technologies, as well as a broader adoption of the successful technologies. We have a couple of environmental drivers that really dictate how we, what sort of projects we fund. First, we're looking at the sustainment of testing and training ranges, as well as facilities and operations. And this means it's very broad. We look at everything from a changing environment to maritime sustainability to dealing with unexploded ordnance and the chemicals associated with them. But we also look at reducing current and future liability. And this would include a traditional pollution prevention program where we're looking at eliminating or substituting chemicals of concern or processes that are being utilized in the manufacturing, maintenance, and operations in the military installations. And then we're also looking at impacts from past practices. And so that would be any chemical of concern that's impacting groundwater, soils, and sediments. And that's where the presentations you'll see today are falling within this area of dealing with the impacts from past practices. We have several focus areas that we address within our programs, including PFAS and AFFF, installation restoration, installation resilience, munitions remediation, sustainable energetics, corrosion and repair technologies, as well as electrical infrastructure and planning. And what you'll be seeing today falls in the realm of PFAS and AFFF. SERDP and ESTCP have a very large program associated with this. And you can't really view this entire slide, but you can go to our website and see this information on our website. This information is interactive. It gives you information on everything SERDP and ESTCP have funded in the realm of dealing with PFAS and AFFF in the environment. So I really encourage you to take a look at this. If you can get to our website, this will be linked there. Integral to our program is the technology transition of the work that we fund. We look at all different ways of getting the information out to people, including videos, guidances, manuals. We have an EnviroWiki page. We do in-person training. And then ultimately, we do the webinar part. And this is really important to us so that we can transition this information to you. The webinars here are coming up over the next few months. All of the information on these is available on our website at this point. And you can see we've got a few coming up that fall within the environmental restoration area, as well as another PFAS webinar towards the end of the calendar year. If you go to this site, you can see the list of all of the webinars we have coming up. And you can also get the archived webinars from past webinars that we've done since the inception of this program. So there's a lot of good information there. And before I turn it over to our presenters today, I also wanted to mention the SERDP, ESTCP, OECIF, and OEPF symposium that will be held in Arlington, Virginia. Registration is open for this. This symposium highlights all of the technologies that are being funded by these programs, including SERDP and ESTCP. It's an excellent way to get information on some of these cutting edge technologies, and I encourage you to take a look. So at this point, let me turn it back to Rula to introduce our speakers. Rula?
Thank you, Andrea. It is now my pleasure to introduce our first speaker, Dr. Rainer Lohmann. Rainer is a professor at the University of Rhode Island's Graduate School of Oceanography. His research focuses on the detection, fate, and transport of anthropogenic pollutants in the environment, often relying on passive samplers in the process. He is the director of an NIEHS-funded URI Superfund Research Program Center on the sources, transport, exposure, and effects of PFAS. The center is referred to as STEEP. And since 2000, Rainer has published over 180 papers and book chapters. He is a member of the executive committee of EPA's Board of Scientific Counselors and serves as editor of Environmental Toxicology and Chemistry Journal. Last but not least, he received a degree in chemical engineering from France and a doctorate degree in environmental science from Lancaster University in the United Kingdom. Rainer, we're very pleased to have you. Please get us started.
Thank you very much, Rula, Andrea, and thanks for inviting me. And good morning, good afternoon to the attendees. So I'll talk about passive samplers, and in particular, why or how they can help us with PFAS. And then I go into three examples. One is an equilibrium sampler that we developed with funding from SERDP, a graphene hydrogel sampler, very small and quick. And the second is a kinetic sampler, so a sampler that integrates PFAS over a longer period of time, and in this case, the polyethylene tube. And then lastly, there's a little side exercise that we perform to make sure we actually get good results for some of our solvent choices for collecting PFAS. So with that, let me get into some details. So first, what is a passive sampler? And passive samplers are very useful tools as they collect or reflect the chemical activity of chemicals, compounds that are either dissolved on the gas phase. And by chemical activity, I mean they only can measure what is available to diffuse into the sampler. And that is basically the active part of the chemicals and the one that leads to bioaccumulation and transport. And the beauty of that is that typically these passive samplers have some outer membrane, maybe it's an entire polymer, and they typically are not capable of taking up particles. So we really only measure what is dissolved. And that certainly helps quite a bit. Another benefit that we often gain from using these passives is that we actually have a time-weighted average concentration during the entire deployment period. And that certainly helps us figure out what is a realistic concentration they would worry about on average. And that might be very useful if you're worried about surface water concentration that might lead to contamination of fish and fish consumption advisories. I think the other part that we've realized, particularly for PFAS, is that the samplers help us actually do the pre-concentration step in the field. So typically, if you do a water extraction, you often take it to the lab, you filter it through a cartridge, and then you extract the cartridge. In this case, we basically take this cartridge into the field. And so that helps us with less work in the lab, less interference from other matrices. But of course, lastly, this is not free of charge. Otherwise, SERDP wouldn't pay for us. And many other investigators, we have to fully validate and understand how these samplers work. So that involves either knowing the equilibrium partitioning constants if we're looking at an equilibrium sampler, or we have to understand something about the sampling rate, so what volume of water is extracted by the passive sampler over a given time, and how are these affected by environmental factors. So with that said, let's do a quick equation so everybody's happy. So the general equation to describe the uptake is basically the amount taken up by the sampler is a function of the dissolved concentration in the surrounding water, the partitioning between the PFAS between the water and the sampler, and the mass of the sampler we deploy. And then there's a term that basically describes the kinetics. And if we take, do a really short deployment, we try to stay in the linear uptake. So in the graph on the bottom right, you see initially the uptake is basically linear. The fusion is always going in. And for a certain amount of time, we can actually proceed and assume that the uptake is always the same amount of water filtered. And that's the linear uptake phase. If you wait long enough, we reach an equilibrium, a plateau ideally, and here the amount in the sampler simply depends on the concentration in the water surrounding the sampler and the equilibrium partitioning constant. So those are the two extremes, and we really like to say either in the linear uptake or in the equilibrium side. All right, so the first sampler that we developed, and that was through SERDP funding collaboration with colleagues Bob Hurt from Brown University, is a nanographene. So a suspension of, and you can see this on the right, there's some pictures, there's a brown solution, and that is basically graphene oxide, and that basically then gets converted through a Hammers method into a cylinder that is basically black from a lot of carbon. But if you pay close attention, you see in the top right, a very, very porous structure. So this nanographene is mostly water, but a lot of pore space, a lot of active surface area. So it's a very good condensed way of having a lot of surface area for sorption. And you can see it on the right next to the coin is actually a fairly small unit. And this is for us is what we call a quick equilibrium sampler. And you can see it here in action. You can see a pin set, you see the remnants of 2 fingers. So it's a fairly small device. And here we do a series of lab experiments. And when I say we, it's mostly Dr. Jitka Besanova, who has been leading this effort for us. So if we then had this nanographene synthesized, she did a series of experiments to understand the partitioning, so the affinity of the different PFASs to the nanographene. And this is shown in this graph. It goes from the left with 4-carbon carboxylic acid, PFBA, all the way to tetradecanoic acid with 14 carbons. And then you have in red several sulfonic acids, in green some sulfonamides, and then GNX and a fluorotalumide sulfonate. And you see the partitioning increases as the molecules become longer in their tail, which is driven by hydrophobic interactions. But the attraction to the nanographene itself for PFBA is not very high. So once we realized that, we went back to the colleagues at Brown and said, look, we have to improve the surface because it's not being attractive enough. And so the beautification or the increase in utilization of this nanographene was through some chemistry. And so in this case, the group at Brown grafted on an amino-benzylamine to change the overall net charge on the surface from being negative, which led to the repulsion of the short chain anionic PFAS, to an attraction. So we basically created an electrostatic attraction for the charged, the anionic PFASs. And so you can hear the reaction scheme, but the key part is that we managed to switch the overall net charge from being negative to being positive on the surface for anything from pH 3 to 7. So with that chemical modification, we now arrive at this situation where you see the same graph as before, but you now see 2 bars for every PFAS of interest. And the left-hand side of each is always higher by roughly an order of magnitude. So the partitioning to the nanographene increased through this modification of the chemical surface by roughly a factor of 10 for almost all of the carboxylic acids. So you start seeing PFBA, and suddenly the increase partitioning is 10 to the 2, and it goes all the way to 10 to the 4 or 10 to the 5. You see a similar increase for all of the sulfonic acids, roughly a factor of 10 more attractive. This sulfonamides, there's no great change because they were not driven by electrostatic, but mostly by hydrophobic interactions. And you see a similar increase for GenX and the fluorotalumide sulfonates. So that's basically our current version, the surface modified nanographene. The exposure we do is 24 or 48 hours. The extraction is very fast. So as I said, this for us is an equilibrium quick sampling tool. We then took it to the field. In this case, we went to the Delaware River. Delaware River has plenty of fluorochemical industries along it. And we thought we could see whether our samplers work reasonably well. So we performed a combination of taking grab sample, deploying the samplers for several days, and then taking another grab sample and comparing with the results, which you see here. The top part of the graph is the concentrations of different PFASs in the water from grab samples. And the bottom is the same compounds in the graphene hydrogel. And the good news is the profiles are very similar. The bad news is that in this initial deployment, we're not doing that well with understanding all of the environmental factors impacting the work of the nanographene. So while the grab sampler recorded tens of nanograms per liter, the graphene reached just up to 10 nanograms per liter. So we realized we had to go back to perform some more experiments, and that's what we did. The first was the question of whether the influence of salinity or salt will affect the functioning of the sampler. And so the graph on the right, top half of the screen, basically shows partitioning to the nanographene if there's no salinity, fresh water in light blue, half salinity as compared to ocean water and full ocean water in red. And you see the sampler works really well in fresh water, but once we have ions present, saltwater ions, then we start having a problem. And the accumulation on the nanographene drops by almost an order of magnitude. We then also checked whether this depended on whether the cations were monovalent or bivalent. That didn't make a difference. So, all right, first note here, fresh water is easier to work in than salt water. The second part was then any other factors we should know. We were aware that pH will have an effect because it affects the functional group on the modified surface of the nanographene. So at pH 4 or 6, it is a positively charged surface, but as we go up in pH, it loses the positive charge and then it becomes less attractive. You basically go back to having a nanographene that is not functionalized. So second note, we should know the salinity, we should know the pH. Third question was whether the presence of organic carbon, dissolved organic carbon, would affect the sampler, and by and large, it's really not a major effect. If there's a high amount of DOC, it seems to decrease the affinity of the PFAS for the sample a little bit, but at lower DOC concentrations, as you would find in groundwater, that is not affecting the sampler. So that was our quick equilibrium sampler. The next we worked on, that was working with a tool that was developed or first pioneered by Sarah Casason and Jochen Müller from Australia. You can see this in the bottom right picture. Christine Gardner, the students who did the field deployments in Narragansett Bay, and in the jar she has several of these polyethylene tubes. They are roughly 3 inches long, 7 centimeters, and we used them for field deployments across Narragansett Bay. And you see here that we can reproduce the results with different samplers in the order of 15 to 20%, which we think is reasonably good. The testing we did to validate the uptake, as in the sampling rate, was we performed this in a wastewater treatment plant effluent, and we assumed that the effluent would be more or less stable over several weeks. And you can see that on top left, these are every other day grab samples, integrated 24-hour samples showing a fairly steady concentration of different PFASs in the effluent. And on the bottom right, you see the linear uptake over 4 weeks from day 2 to day 28 for different PFASs. And there's a nice linear trend, so we can use them, these samplers, for integrating PFAS concentration over several weeks. We actually did 2 deployments side by side in 2 different treatment plant effluents in Narragansett Bay. And what you see on the graph on the left is based comparison of the sampling rate deduced from one applied to the other. The blue line is the 1-to-1, and you can see that almost all results are within a factor of 2 or better of the average grab sample results. So we think these samplers work well to give us a good estimation of time-integrated concentrations of PFAS in the water. Now, for those that are connoisseurs of passive samplers, I threw in the graph on the right. If we pay close attention to the initial parts of the sampler deployments, we actually notice that very early on we have a higher uptake, and that has to do with the PFAS actually adsorbing to the surface of the membrane before they start diffusing through. So we actually have basically a 2-phase. On the very short time scale, adsorption plays a role. On the longer time scale, it is all diffusion through the membrane to the solvent. We then took the sampler into the lab. This was again part through SERDP funding, and put them into an aquarium, a tank, where we were able to control temperature and flow rates. We wanted to make sure we understood the influence of external variables on the performance of this tube sampler. And here is a little cross-section of the sampler. You see this, the polyethylene membrane, and inside we use a commercial solvent like a HMB or a wax or something of that kind. The uptake is basically characterized as a function of the time it takes to move the PFAS through the aqueous boundary layer, the absorption to the membrane itself, diffusion through the thickness of the membrane, and then absorption to the solvent itself. And we just wanted to make sure that we understand which terms are rate limiting and thus in charge of the overall uptake rates. So here are 2 results for looking at carboxylic acids and sulfonic acids, and basically these are results showing uptake up to 6 weeks. And you start seeing that as an increase over time for up to 6 weeks without a problem. So that's what I meant. We can use these samples to integrate over time scale of weeks. Of course, the question is, how much do we have to know about the conditions outside of the passive sampler? So the water, at what flow it operates. And basically we end up that there's 2 regimes if it's deployed in the groundwater, that's the top part of this graph, the sampling rates are on the order of 5 milliliters per day. And that roughly mirrors what Kazason found in groundwater, what we observed in our tank experiments, and what we basically come out with predictions based on the modeled equation to understand the uptake of PFAS by the sampler. So for very slow flowing environments based on groundwater, we have one set of sampling rates that is just below 5 milliliters per day. If we go to a faster flowing water body, and that's really anything stream, lake, effluent, of course, once we're above a centimeter per second, we're in another regime where basically the exact flow doesn't actually matter anymore, because at this point, the barrier of the aqueous diffusion layer is gone, and it's all dictated by the thickness of the polyethylene tube. And at that stage, we have higher uptake rates, and they're on the order of tens of milliliters per day. Of course, some of you who are working in the field are probably aware of one of these nagging problems. What happens if your sampling tool gets biofouled? How does that affect uptake? So in this case, Matt Dunn, who was working on this for his PhD, took a couple of these polyethylene tubes, let them biofoul in the natural environment, removed the PFAS, and then reused them as a new biofoul sampler. And basically, you can see that the two bars are basically for equal height in every case. There's no effect of biofouling on the uptake rates. We added another term to the uptake equation, and basically, as long as the biofoul layer is not very thick, it doesn't change the controlling factor being the membrane's thickness. So good news is biofouling does not really change the understanding of uptake rates by this passive sampler. All right, and now I'll digress a little bit into what started off as being a slightly more academic exercise. New grad student in my group, Jared Snook, was trying to measure the partitioning of different PFAS to commercially available sorbents. And so he started looking at the literature, what kind of ratios he should use in terms of how much sorbent, how much water, how much methanol to add, and what concentration he should spike. And he realized that the literature was all over the place. The ratios, the methanol content, the concentrations vary by orders of magnitude. So he started off by looking at these 4 different PFASs, sulfonic acid, carboxylic acid, sulfonamide, and fluorotellumer and started playing around with these different 3 conditions that you could have and see if there was an effect on the results. So in the first case, he basically looked at how much water to sorbent he should have in the experiments to not have bad results. And so the graphs on the right basically show you an oasis wax on the left and oasis HLB on the right. And to the left of each of those panels is a blue triangle. That's the reference state, what we think of as the best available partitioning constants. And then as you increase the amount, of sorbent or decrease the amount of solution relative to the sorbent, you start seeing deviation from good partitioning constants. And that is basically once you have too much sorbent and too little water, you deplete the water column and you cannot measure the PFAS necessarily. And so now you suddenly have to start doing a method detection limit replacement. You start introducing uncertainties. And so clearly it's important to have enough water relative to sorbent to get good results. The second step was to figure out what kind of concentration should be added to have decent results. And the problem here is that if the concentration of the PFAS is too high, then you can start saturating the sorbent, and then suddenly the partitioning the partitioning numbers are off. So you can see this here again, as you increase the concentration relative to what is a good amount, you start seeing decreased partitioning constants. So you want to shy away from too high concentration. Jared's conclusion was you want to back calculate, figure out how much do I need in the water to detect it, and then work backwards to not overload the system. And then the last question was how much methanol can you add without introducing a bias? Methanol is typically the phase that's carrying the PFAS into these experiments. And basically in both cases, 0.1% of methanol, the overall system is completely safe. Once you reach 1 or more % methanol, you start having a third phase effects and the partition numbers start to decrease. So with that, he was then able to derive optimized partitioning constants. The paper is published, and he also added a spreadsheet as part of the paper that actually people can use to calculate what is the best way of running these experiments. So let me wrap up. We think passive samplers offer benefits even for compounds like PFAS, which are much easier measured directly compared to some of the other chemicals that DoD had concerns with. The graphene hydrogel we think of as a dipstick. It's a quick tool that could be used to measure pore water or what is dissolved in soil water fairly quickly, of course, also in water directly. The polyethylene tube, on the other hand, is a tool for integrative concentration. So it's something that's best deployed over several weeks. And then you just get one number that reflects the entire deployment span. And the last part, of course, is make sure you know what you're working with by getting the right numbers. And with that, benefits that we think DoD can harness from work like ours, but of course they found a lot more scientists work on passive samplers, is the pre-concentration step that's already happening in the field. It leads to a faster turnaround in the lab. That we good screening tools for site characterization, particularly by only measuring what is actually available, being dissolved. And hopefully down the road, of course, we want to link this to bioavailability, and if things work out well, of course, also then to predict bioaccumulation. And here are a couple of the references that I reported on. And at that stage, I believe I'll hand it back over to Rula.
Thank you so much for a great presentation. Just a reminder, questions can be submitted in the Q&A box. We've received a number of questions for you, Reiner, so I'll start relaying them. The first one is from Woodard and Karan, and it is along these lines. You considered calcium and sodium in the hydrogel sorption experiments. What about interference from common anions, since the PFAS are generally anionic at environmental conditions?
Thanks. It's a good question. I think we look both at chloride and at sulfate. And I'm pretty sure that in this case, the exact makeup didn't make a difference.
Great, thank you. This next question is from the North Carolina Department of Environmental Quality. Do the passive samplers address colloidal transport? And if not, is that a limitation to the method?
It's a good question. I guess it depends on your perspective. So no, the tools, any of the tools that we develop, and I assume almost everybody else that works as a passive sampler, do not work with colloids. Now, is that an important transport pathway? I guess it depends on the site, but if you wanted to assess that part, you would have to grab active samples, and then you can use the passives as a control to figure out how much of the transport or concentration is due to what is truly dissolved and how much is due to something else, which could be the colloidal transport.
Thank you. A follow-up question from the North Carolina Department of Environmental Quality. Below turbidities of 10 NTUs, how might unfiltered PFAS results compare to filtered PFAS results?
Yeah, we have, for several of our field studies, we have done the comparison. And yes, there is some amount of bias or sorption to solids. So again, it depends on, I guess, on the question you're asking and how do you want to address it. If it's about, let's say, transport within a river, not knowing the exact river in question, but if it's fairly simple to do the math, figuring out how much turbidity is present, how much is transported out relative to the water volume. I assume in most cases, given that most PFAS are binding to solids, but not very strongly, I think in most cases, the flux or the mass flow with the water is going to outcompete the solids. I hope that answered the question.
Thank you, Reiner. Next question is from the Pacific Northwest National Lab, and it has to do with whether the sorbents that you're using, are they for one-time use, or can they be regenerated or recycled? And then there's a follow-up question to that. Are the tools you described commercially available? And if so, what is the cost?
Okay, so we reuse our sampling tubes. The solvent is extracted, and of course, once it's extracted, it can be reused because the whole point was to remove all the PFAS that we're interested in. Now, the second question is, no, unfortunately, they are not commercially available. The nanographene is really, you would have to contact us and we would have to work with Brown to produce it. The polyethylene tube is a little easier because it's basically a porous membrane that is commercially available. That's how we get it. It's just cut to size. The plugs is nothing magic, and the inside is just a commercial solvent. So while it's not yet off the shelf, it is somewhat easy to assemble.
Thank you so much. This next question is from the U.S. Army Corps of Engineers. Can you elaborate on which compounds poorly sorb to the sorbents? You did have a slide on that, but the author of the question could not see the color of the dots on the screen. So we'll just mention that the slides are available for download. There's a link to the slides in the chat box. But if you could please elaborate, Rainer, on which compounds poorly sorb, that would be great.
Sure, and I'm sorry that it wasn't that easy to see. So the simple answer is similar to what is basically known for, let's say, granular activated carbon, where there's been a lot of research. So sorption is much better the longer the chain is. So the PFNA is doing much better than PFBA. Sulfonic acids are doing better than carboxylic acids. Neutral polyfluoroalkyl compounds do better because they are more hydrophobic, so they are sorbing well. And then we had fluorothiosulfonates, and again, there it was an increase with chain length, but because they're not as acidic, they sorb much better. So basically, the samplers work well for most PFAS, except it gets tricky with the 4-carbon chain PFBA, PFBS, and of course, shorter chain compounds.
Thank you so much. This next question is from Groundwater Technologies. Are there plans to test the passive samplers in variable length monitoring well screens and compare the results to traditional sampling methods?
As part of our SERDP funding, we have comparison with other tools, with traditional approaches, if you wish to use that word, that is going on. I don't think it's particularly in wells right now. In this case, we have some surface water deployments, but yes, so these kind of cross-tool comparisons are going on, and some of that work is facilitated by Mark Mills from the EPA.
Great, thank you. This next question is from the University of Colorado, Denver, and it relates to the differences you described in the tube samplers between groundwater and surface water results. Do you suspect that this difference stems from the difference between laminar and turbulent flow, respectively?
Yes, great question, and you've already given the answer, yes. So we think that, and maybe I didn't express this very clearly, the tube sampler was designed to actually be a flow independent sampling tool. So we could use it in basically one sampling rate for all environments. And that works as long as you're not in a place like a groundwater where the flow is so slow that you actually have an aqueous boundary layer that is thicker in a way and slower for the PFAS to get through than the membrane itself. So yes, the laminar flow is causing the problem in the groundwater situation.
Thank you. This next question is from ECC. Are there any competitive interactions between various PFAS that change their absorptive behavior onto the graphene?
Excellent question. So I didn't want to get into this detail. So there's two possible reasons why we didn't do very well with our first deployment in the Delaware River. And one is that we obviously underestimate some of the environmental factors impacting this nanographene. The other part was that the Delaware River is actually known to have a much wider range of PFAS present because it has fluorochemical industries upstream. So it could have actually been an out competition on top of everything else. And we are looking into that right now too. But as I've shown, the main results from the environmental exposure already tell us that knowing exact pH and salinity is going to be very important.
Great, thank you. When do you expect those results to be available, Reiner? And when do you expect to have a final report available for download on the project webpage, which is the last slide in your presentation? When do you want us to refer people to your project website for a final report, roughly speaking?
I think Andrea's given us another year. As always, everything takes longer than we anticipated, but I guess the key part is the main publications are out and the other ones are coming forth in a prompt manner, academically speaking.
Academically speaking. Well, thank you. And as a reminder for everyone, Reiner's contact information is available on his project webpage as well as in the PDF of the slides. Okay, we have time for a few more questions. You may have referred to it, but it's come up a few times from different individuals. How do you measure the concentration of PFAS inside the samplers? Do you sacrifice them? Can you go over that again? There's lots of interest in that, Reiner.
Sure. So I assume the question is, so on the nanographene, unfortunately, it's a one-time deal because in the process of the extraction, we destroy it. So that's an easy one. For the polyethylene, so as I was trying to show, it's a porous tube. It has two caps, and inside is just solvent. So for the extraction, we actually take out the solvent, extract the solvent, extract the tube, and then we can reuse them. Yes, and then we can reassemble. So we start reusing them, and we just want to. We're just a little more careful if we had a really high concentration deployment, like in a AFFF plume, then we separate those out because we don't want to have problems. But other than that, for general deployments, we can reuse them.
Great, thank you. And we've also received a number of questions around detection limits for these samplers, and also whether the methodologies or the tools are EPA approved. So if you could address the detection limit issue and then perhaps elaborate on how you can use these tools together with other tools that are EPA approved, that would be great. We've got about 4 to 5 questions along these lines. Okay,
so if I talk really slowly, I can dodge the EPA question. Detection limits for the tube sampler are in the low nanograms per liter or below. So it's a sensitive tool. The nanographene is a little higher just because the actual mass of carbon we have in that sampler is fairly small, which is both benefit and, I guess, challenge. But of course, a lot of the work that DoD is interested in is at slightly higher concentration sites. So detection limit haven't really been a problem for us in most situations. And the question for EPA, so I have to operate a little bit with blinders because for hydrophobic contaminants, EPA did a lot of work to facilitate the inclusion and guidance documents for how to use passive samplers within EPA methods. And so that approach was very powerful and very useful and led to huge acceptance. We're not quite there yet with PFAS passive samplers. I think that's partially because a lot of the work obviously is just being funded by SERDP, and we haven't yet coalesced around the best tools and the best approaches. So I assume it's because the work on the hydrophobic chemicals has already been done. The acceptance will be a little easier. And of course, given this funding that SERDP is investing in several projects like mine, that will also help. But it's right now, we would have to squeeze in through 1633 or something. But there's no specific method for a passive sampler at this point, as far as I know.
Great, thank you. And the last question for you is from the US EPA. Can you discuss possible cross-contamination for normal PFAS sampling at groundwater sites? There's the potential for field reagent blanks. Is there a similar approach for blanks when relying on PFAS passive samplers?
Yeah, so for all experiments or field trials, we have a series of blanks as in samplers that were prepared the same way that we just either have in the lab as blanks or take to the field as field blanks. So we have all of that and we correct if needed. But because this is all under our control, we really typically we don't have any major problem with blank contamination.
Thank you so much, Rainer. We did not get to all the questions, but if there's time towards the end, we will get back to you. At this point, we're going to go ahead and transition to our second and last speaker today, who is Dr. Craig Devine. Craig is a national technical expert and senior vice president at Arcadis. He has over 25 years of experience characterizing and remediating a broad range of contaminants, utilizing a wide range of technologies. Craig has authored and co-authored over 50 papers and is an associate editor for Groundwater Monitoring and Remediation, as well as the Remediation Journal. Finally, he received a bachelor's degree in biology from Wheaton College, a master's degree in watershed science from Colorado State University, and a doctorate in geochemistry and hydrogeology from the Colorado School of Mines. Craig, welcome. Please go ahead.
Hey, thank you, Rula. Okay, today I'll be presenting on the vertebrae segmented horizontal well technology, specifically for monitoring PFAS mass discharge over time. And here's an outline of the presentation today. First, I'll start with an overview of the vertebrae system. Then I'll describe the field site and the specific vertebrae construction details. Then we'll review a comparison of the various groundwater flux measurement methods we tested with this system. And I'll talk about the mass discharge analysis. And finally, I'll close with some lessons learned, conclusions, and benefits for the DoD. So simply stated, the vertebrae system is a segmented multi-port horizontal well that was developed by a company called NREX. And as you can see in the picture in the upper right, each screen, which is depicted in green and red, is separately plumbed to the surface and separated by a grout seal. The picture on the bottom left shows several color coded risers, and each of these is connected to a different screen. An individual vertebrae can be installed with more than 20 screen segments for the whole system. And the entire system, including all these screen components and risers, is actually constructed off-site prior to drilling activities. And then it's shipped to the site on these large spools that you can see in the picture on the bottom right. So at this point, it's important to note that because the well is designed and constructed prior to mobilization, you must already have sufficient geologic information to provide the basis for your well design. And also, because the well is installed using a mud-based system, there's very little additional geologic information that's obtained during the well installation. Like other horizontal or directionally drilled borings, the vertebrae system can be installed under surface infrastructure like buildings or flight lines. And I think this is a key feature. In a comparative sense, these wells are relatively inexpensive and in fact become more cost-effective than conventional vertical monitoring wells once you start having more than 6 or 7 screens per system. And of course, that's site-specific, but once you get to these larger systems, they become very economical in dollars or cost per screen. Also, I'd like to note that this technology has actually been around for more than a decade, and there are over 200 wells that have been installed to date. However, this technology has not specifically been used for PFAS contaminants or for monitoring PFAS mass flux or discharge. And that's really the focus of this work. The field site where we deployed this technology and demonstrated it was at the Grayling Army Airfield in northern Michigan. At this site, total PFAS concentrations in groundwater immediately downgrading to the source area exceed 30 micrograms per liter. The site is sandy and quite homogeneous, with hydraulic conductivity of most samples falling in a relatively narrow range between 0.01 and 0.1 centimeters per second. I see a typo on the slide there. It should be centimeters per second, so this is a very permeable setting. The site was selected in part because we could build on efforts from a previous ESTCP project led by Joe Quinnen, where PFAS mass flux was characterized using high-resolution site characterization methods that included hydraulic profiling tools, or HPT, vertical aquifer profile, or VAP sampling, and a mobile on-site lab for PFAS analysis. And the results of this previous work are shown in these oblique cross sections in the bottom right, which depict the spatial distribution of PFAS mass flux along 3 transects, and the warmer colors represent higher mass flux values. So the methods used in this previous project allow a detailed snapshot, but this snapshot is a single snapshot in time of mass flux distribution. However, the key goal of this current project was to look at the vertebrae system to install a permanent infrastructure that could be used to monitor changes in mass discharge over time. As I mentioned before, the vertebrae technology has been around for several years, and so the installation methods are pretty well established. Photograph A shows the relatively small low mass drill rig and the entry pit. You can also see the mud in the pit, which is used to bring the cuttings back to the surface and keep the borehole open. Photograph B shows the asymmetric drill bit that is used to cut and steer. Photograph C highlights the real-time navigation system. A transponder is located immediately behind the bit that emits a signal which is detectable by a walkover navigation system. Photograph D underscores the control and accuracy of this system. It shows the drill bit exiting the surface about 500 feet away from the drill rig. And as you can see, the drill bit actually hit the stake with the orange flag, which we had used to demark the planned exit location. Once the pilot bore is drilled, the bit is replaced with a reamer bit shown in photograph E to enlarge the hole and further remove cuttings. Once the borehole is adequately reamed and the prefabricated well system is daisy chained to the bit, as shown in photograph F, and then pulled back into the borehole for installation. And at this point, grout is then pumped into various tremie pipes that are installed between each of the screen segments to create seals between the screens. After this, the well is developed using a variety of techniques to enhance communication with the aquifer and remove residual drilling mud. So at the site, a total of 3 vertebrae well systems were installed, which are depicted in the aerial photo on the left. 2 wells, a shallow and deep well, were installed in a transect configuration running approximately southwest to northeast. And these wells are also shown in the cross-section figure on the right. And their alignment is overlaid on historical concentration data, which provided a basis for the well position and the screen locations. You can also see that there's another well installed roughly north to south, starting at the building 1160 source area and follows the direction of groundwater flow. We've termed this well a long sect well and are using it to look at changes in PFAS concentration and PFAS species distribution along the groundwater flow path. However, in the interest of time, today I won't be discussing the results. The table on the bottom provides some more details on these wells, and as you can see, they're each about 450 to 500 feet long and each contain 8 separate screen segments. The key objective of this demonstration was to verify the placement of these vertebrae wells, and specifically the question was, can we place them where we want to, and are they where we think they are? So to answer these questions, we actually compared several sets of coordinate data, which I'll show in the next slide. Specifically, we have the target boring profile elevation. These are the locations where we wanted to place the screens. We then compared that profile to the navigation data collected during drilling. This is the data that tells us where we think we are during the drilling activity. We also placed an RF tracer line in the boring and came back after the well was installed and used conventional geophysical techniques to develop an independent depth and location data set. In short, we found very good agreement between all 3 coordinate data sets. In fact, 6 of the 6 screens that were located along the horizontal section of the shallow transect were within 1.5 vertical feet of each other. However, as you'll see in the next slide, the 2 screens on the end had more variation, and we think that this is because the transponder is less accurate when the bit is angled during the drilling activity. So there's more uncertainty about where you are. So a key takeaway for us was in the future to avoid placing any screens on a slope, and that'll help improve our confidence on location. I'm having difficulty moving forward here.
Craig, just go ahead and we'll address the issue.
Okay, well, I'm sorry, I seem to have actually even lost my screen here. All right, well, I'll proceed. Let's see. Yeah, I'm sorry, Rula. I have printouts, but I don't know I have the equivalent slide number. So I'll proceed.
We should be getting the slides up in just a second. So if you go ahead and continue from
Okay.
and I can help you with the slide numbers if you'd like.
Yeah, sure. Okay, so the slide that'll be coming up is and I guess, sorry, Rula, one more question. I don't see slides, but are other folks seeing slides?
Nobody's seeing slides right now.
I'm not sure if it's just me that's not seeing slides.
It's not you. We're on slide 55. And we're waiting for our host to reshare the slides in just a moment.
Okay, no problem.
If you'd like, I can pause for a minute and start relaying some of the questions.
Sure. Yeah, let's do that. Yeah, that's good.
This question is from Leidos. What sampling rates or pumping rates are possible with the vertebrate well system?
Yeah, that's a good question. I will say for the purpose of this project, we weren't looking at the vertebrate well for anything other than conventional sample collection. And to some degree, that's of course going to be dependent on the formation. However, these screens are relatively small diameter and they're wrapped in a geotextile membrane to act as sort of a filter pack. And so there is a kind of a practical upper limit. I would defer to the experience of the folks at NREX to get more information about pumping and injection rate. I know they've used these systems to inject remediation fluid, to extract water, and also and even SVE type applications.
Wonderful. And if you share with me really quickly the information of the person you work with at NREX, I'm happy to share that with the attendees. But
yeah, sure. I'll give his contact information. His name is Lance Robinson at NREX, but I'll provide the contact information.
Wonderful. Looks
okay,
like
I'm seeing the slides here.
Wonderful. And if you could please accept control and go ahead and restart.
Yeah, you bet. Okay, I'm sorry everybody for the little hiccup there. So here are the data I mentioned on the previous slide. The yellow series is the ground surface elevation. The gray series is the target elevation. So that's where we wanted to put the hole or wanted to put the screen. The blue series is the transponder elevation that's available during drilling. And the orange series is the elevation data from the geophysical testing completed after the well was installed. So visually, you can see the consistent alignment in the horizontal section of the well. And you can also see these deviations at the end of the well where the well path is curved and the screens actually are installed at a slight angle. And the various labels, for example, S-P8 represent the screen names. In this case, S is shallow. I think P represents purple and 8 is the 8th screen. Another important objective of this demonstration was to assess the placement of the grout seals between each of the well screens. So as I mentioned, the well is installed with tremie pipes located between each of the well screens, and the grout is pumped into the tremie pipes to create a seal between the screens. This is shown on the lower part of the lower figure, where the green bars represent the screens or monitoring intervals, and the gray bars and dots represent the planned grout intervals and the tremie pipe locations. And the tan zone is where we anticipated formation collapse on the well. To confirm the grout placement, we used a fiber optic distributed temperature system, or DTS, that was installed with the boring. This system is very sensitive, and you can collect spatially continuous temperature measurements at various time intervals. So the figure in the upper left shows the temperature along the y-axis and the well orientation on the x-axis, and compares the temperature profile measured at 2 times, one within about 24 hours after grout placement and another one several days after grout placement. And so you can see these distinct temperature spikes that are associated with grout curing at these locations. And for the most part, grout was detected approximately where we plan to install it. However, as you can see, especially on the right side where the boring angles upward, there are some signatures that overlap with the well screen intervals. So we believe this is likely that grout have flowed down the annulus in these angled sections. This table shows the pH, specific conductivity, and total organic carbon values measured at various well screens for 4 monitoring events over the course of a year. I won't spend much time here other than to note that the values highlighted in yellow suggest the influence of grout at locations near the ends of the well. So from a practical perspective, the main lesson learned here is to make sure the screens are placed far away from the angles on either end of the boring. The performance assessment included 3 main data types and analyses. The first is the PFAS concentration data from quarterly groundwater sampling. The second is the estimation of groundwater flux using several different methods, including pneumatic slug testing, single well tracer testing, and fiber optic active distributed temperature sensing, or ADTS. And finally, these data were used to calculate PFAS mass flux and discharge. And this was northern Michigan, so occasionally some difficult field conditions. This slide shows a comparison of data collected from the vertebrae wells to previously collected VAP sample data. The x-axis plots the various screen intervals, and the y-axis represents concentration and log scale, and the 2 color series are for PFOA and PFOS. The VAP sample data are noted by the X's, and the circles represent the average concentration over 4 quarters from the vertebrae wells, and the bars represent the concentration range from those wells. So overall, you can see very good visual match in that the chemical spatial heterogeneity is preserved, both vertically and laterally. So the main takeaway with this here is that data from vertebrae screens can be considered representative and comparable to VAP data. And furthermore, this implies that VAP data can be used to predict concentrations that will be measured by a vertebrae system, so useful for well design. Next, we'll cover several groundwater flux measurement techniques, and the first is this pneumatic slug testing. The photograph on the left shows the field setup where the casing is pressurized with a hand pump, and then the pressure is instantaneously released with a valve. The graph on the right plots the observed recovery data and are compared to a slug test model. We found that in general, the setup is straightforward and easy to deploy in the field. However, as you can see here, the data deviate notably from the model predictions in both early and late times. So that's sort of an indication that some of our key assumptions are not valid. And there are some key takeaways overall from the pneumatic slug testing. First, the hydraulic conductivity results were more than an order of magnitude lower than expected for essentially every test. And also the interpreted specific storage values were really, really low, really implausible. And as you saw on the previous slide, there were significant deviations between the observed data and the characteristics of the model. So overall, this implies there's some important assumption violations, and most likely we're seeing well skin effects that are reducing the hydraulic connectivity between the screens and the aquifer. And as I think I previously mentioned, we believe this is because these screens are wrapped in a geotextile membrane that is designed to keep sediment out of the well. However, the permeability of this membrane is likely lower than that of these really high permeability sands at this site. So for lower permeability sites, the hydraulic effect of this wrap may not be very significant, but for this site, we believe that it resulted in the slug test data being not reliable and biased low. The single well tracer test method was adapted from well-established methods that we've used for years for vertical wells. Essentially, a tracer solution, a salt-based tracer solution, is recirculated between the well screen and a reservoir on the land surface using a pump. And as tracer-free groundwater flows through the well screen, it dilutes the tracer concentration in this recirculation loop. And that dilution rate is proportional to groundwater flux. So the measured data are then interpreted using a model shown in the figure on the right. The single well tracer test results were within the expected range of the site for most of the tests. And also we completed a repeat test, and it agreed with the original test within about 12%. So I think the method is pretty reliable and repeatable. Also, the tracer dilution data were largely consistent with the model that was used for interpretation. So that indicates that the underlying assumptions are valid. But I would like to note that the interpretation model does require some assumptions about screen permeability and well construction that can't be directly measured. So there is some unresolvable uncertainty and potential for bias with the technique. Also, because we felt like these tests were largely successful, we think there's value in additional work. Further optimization could probably improve the performance of this method. For the ADTS method, the fiber optic cable that's installed with the well is heated and then allowed to cool. And in a way that's somewhat similar to the single well tracer test method, the measured cooling rate is used to estimate Darcy flux. However, in this case, the analysis requires an empirical relationship that is developed through laboratory methods that use site soils. And you can see that essentially kind of empirical calibration curve in the bottom right. Overall, we felt the DTS method performed quite well. The results were reasonable and exhibited spatial variability. The graph on the right shows the well construction in the colored column with the continuous and average flux values in the next columns to the right. The graph on the bottom left shows groundwater flux for each screen for each of the 4 monitoring intervals that were calculated from this method. So this graph compares all of the results for each of the methods, including some additional methods I didn't talk about, and to make them comparable, because some measure hydraulic conductivity directly and some measure flux directly, we converted them all to hydraulic conductivity using Darcy's law. The x-axis is hydraulic conductivity in log scale, and the y-axis is depth below land surface. And as you can see, far to the left are the results from the pneumatic slug test data. And it's clear that this data are too low and that there's bias there. But for the other methods, while there's some variability within and across these other data types, you can see how relatively uniform these values are and range only over an order of magnitude or so, which is a very low variability for most geologic systems. So we use this to support the mass discharge analysis, and this was completed by discretizing the transect into a series of rectangles where the mass discharge for each rectangle was calculated by multiplying the concentration by representative groundwater flux for each sampling time. These elements were then summed to estimate the total mass discharge across the transect. This table summarizes the total PFAS mass discharge for each of the monitoring events and compares what we consider the 3 most reliable methods: HPT, single well tracer test, and ADTS testing. The average of these 3 methods is plotted on the graph below. And although there is a clear difference between the methods, the range is consistent with the variability in the hydraulic conductivity. So from our perspective, we consider all of these methods useful and reliable. And maybe on a personal note, I think maybe mass discharge accuracy, while it's important, it's probably less important than precision or repeatability, because in most use cases, we are typically more interested in understanding trends in mass flux and mass discharge over time, as opposed to specific values. So a quick side note here. Remember that I mentioned a couple of times the site is very homogeneous and highly permeable. In fact, our team refers to this site as the Borden Aquifer of the South. However, the groundwater dynamics are quite notable. There's very fast recharge. The gradient magnitude varies by more than 50% over the year, and flow direction changes more than 30 degrees. And as many of you know, PFAS accumulate at air-water interfaces, and therefore we can expect complex behavior in a dynamic setting like this. I didn't mention it previously, but we do have additional monitoring infrastructure that is focused immediately near the water table, and we observe very high spatial and temporal concentration variability over the duration of a year. And the point here is that this zone immediately near the water table is likely very important for many sites, particularly with respect to PFAS transport and mass discharge. And so a general recommendation is that this zone should be given special consideration when we're designing monitoring programs. There are a number of lessons learned that I'd like to note. First, the entry and exit angles of the wells should be limited to prevent tight bends, which aid in access to and sample collection. The screens, as I mentioned several times, should be installed horizontally, not at angles. Also, we've noted the geotextile screen wrap may reduce hydraulic connectivity, particularly in high K settings, and maybe less of an issue in low K settings. Finally, we'd like to recommend dedicated sampling tubing for each of the well screens. A few conclusions. First, the vertebrae screens can be installed within about 1.5 feet of the target depths with confidence for similar settings. And personally, I consider this an amazing capability and a great advantage of these systems, the high degree of control and accuracy of placement. DTS is effective for confirming the as-built grout seal positions. PFAS concentrations from vertebrae wells can be considered representative. ADTS and tracer data yield reasonable groundwater flux values. Even in simple settings like this, there's significant spatial variability. In fact, we found that more than 90% of the total mass discharge for that cross-section I showed was sourced by only 2 of the subzones. So this implies the need for relatively high data density and resolution, even for simple systems. Mass discharge ranged about 40% over the year. And so this also implies that relatively frequent monitoring is necessary to capture this variability, probably quarterly for most settings. And finally, the capillary fringe and zone near the water table is likely an important area for PFAS transport and mass discharge at many or most sites. We consider this a very successful demonstration, and personally, I find this a very exciting technology. I'll close with what I see as some key benefits specific to DoD. First, vertebrae is commercially available and is cost effective, and as I mentioned, it's owned and well demonstrated and offered through a company called NREX. It's easily installed, including underneath active surface infrastructure. It allows the measurement of mass discharge over time using methods that generate consistent, repeatable data. Mass discharge monitoring can support remedy evaluation, risk assessment, and other objectives. At this point, I'm happy to field any questions.
Wonderful. Thank you so much, Craig. We do have a number of questions. The first one is from the South Carolina Department of Health and Environmental Control. Is there a maximum length for the well?
Yeah, that's a great question. Again, I would defer to the specific NREX experience, but I would say, for typical geologic settings, a well that's 1,000 feet long with 20 screen segments is doable for most settings. Longer may be possible, but these can go quite long.
Thank you, Craig. This next one is from the Michigan Department of Military and Veterans Affairs. Do you think the drilling mud coated the well screen and impacted the observed hydraulic conductivity that you reported?
Yeah, that's a great question. Just in general, mud-based systems or mud-based drilling, offers that challenge. In this particular case, we used basically a biopolymer mud that is biodegradable, and we used an enzyme breaker to facilitate that degradation. So I think, of course, the development activity, you look at changes in yield and a number of different measures to confirm that you've got good hydraulic connection to the aquifer. We did look at organic carbon over time and in the wells, and in a number of the screens, we did see relatively high values, especially in the first couple samples, but over time, those values decline. So I would say, I think that risk is manageable, especially if you're using a biopolymer as opposed to like a bentonite mud.
All right, this next question is from EPA. And it's really just asking your opinion about whether this is a good tool for what is being proposed. So let me relay the question. You'll see what I'm trying to say here. Some people are proposing to develop soil cleanup concentrations based on mass discharge values. You said mass discharge is good for trend analysis using the vertebra wells. Do you think and support the use of deriving soil cleanup values using mass discharge with your tool?
Yeah, that's a good question, sort of about the philosophical use of mass discharge in plume management. I don't mean to be vague, but I do think it's depends on the site specific situation. I think in some cases, a remedy objective around a specific mass discharge value may be supported. Probably in most cases that would be derived through a risk analysis that would develop that number, and that provides a quantifiable target for performance objectives for a remedy. I think in most cases, though, having an analysis of mass discharge helps us understand the changing risk profile of the plume over time, because that represents the mass that's moving. So that can give us sort of a long range complement to plume footprint maps that come from conventional concentration to understand plume dynamics over time. And it can also be used to help us understand the benefits of various remedy actions. You know, for example, if there's source removal or some other remedy action, you can look at the impactful effect of that over time. And in some cases, it may even guide you to make further changes. So I guess a bit of an open-ended answer to that question. I do think in some cases, target may be appropriate, and probably in most cases that'll be driven by a specific risk assessment. In other cases, I think it's really just useful to understanding plume dynamics, changing risk profile, and the benefits of various remedies or partial remedies.
Thank you. This next question is from the Washington State Department of Ecology. How were the risers installed in each of the individual wells?
Yeah, so it's kind of an interesting technology in that the whole thing is fabricated ahead of time. And if you maybe go to slide, it's titled Overview of the Vertebrate System. It's my 5th slide. I apologize, I don't have your slide number. Or sorry, it's my 3rd slide. You can see, you can kind of get a feel. Yep, just passed it. Yep, that one right there. You can kind of get a feel for it. Whoops, one more. It's got the picture of two back. There you go, right there. All right, so you see the conceptual diagram of the screen, and then the green, in this case, the green screen, and then a green riser, and it goes through that grout and then up to the surface, and the same thing with the red seal. And when this well is constructed, those screens are emplaced in a sheath, which you see on the spool to the bottom right. So all those screens and risers are already put together and then spooled together on that sheath. And then when the well is pulled into place, that whole system, that sheath included, gets pulled into the borehole. And you can see that on the picture there on the bottom left. And then that sheath is actually removed. So everything is kind of protected by that sheath during the install, and the screens and the risers are already pre-constructed and built. And they're also, you can't see them here, but they're also tremmy pipes for the grout that go between each of these screens. So there's actually quite a number of pipes and tubes that come out of each end of this hole that represent the risers and the tremmy pipe locations.
Great,
I hope that made it a little bit more clear.
Thank you. A follow-up question from the Washington State Department of Ecology. What are the depth limitations for installing these wells?
Yeah, I mean, there's really no practical depth limitation. It's more of a length limitation. So let's say that 1,000 feet is sort of the practical limit. You do have a setback distance. So, it varies on condition, but let's say your setback requirement is 1 to 5. And I think probably most settings you can be more aggressive, but the math is easy. If you want to go 20 feet deep, you need to step back 100 feet, 20 times 5. So that's sort of the depth limitation. If you're going deeper, you need to step back further to get to depth. Now with these smaller systems, I think the angle can be a little bit more aggressive, but that's maybe the depth is more controlled by the length. So I will say, I've got personal experience installing horizontal wells to maybe about 130, 140 feet. And, for most unconsolidated systems, we don't need to go deeper than that.
Thank you. This next question is from the Colorado School of Mines. Assuming the slug test is done through the sampling pipe, do you observe any air trapping during the test? Will that
that could be an issue as well. We did 2 types of slug testing. We did this pneumatic test where we pressurized with air and then released that pressure and looked at the head rise. We also did do kind of a conventional slug test where we added water to the riser. And in both cases, I would consider the results highly suspect. So basically, I don't think these systems are well suited for slug testing. And there's also kind of a complex geometry. Conventional wells are vertical, and a lot of our solutions are well suited for interpretation of that kind of vertical geometry. Here we've got a horizontal well, and in some cases it's not far from the water table surface. So long and the short of it is I wouldn't recommend slug testing for these systems.
Thank you. Lots more questions coming through. I'm gonna maybe have you answer 2 more, Greg, and then we'll wrap up with a few more questions that have come in for Reiner.
Sure.
So really quickly, this next question is from the U.S. Army Corps of Engineers. Given the acknowledged variability in the test environment, would this tool be discouraged for use of shorter term source identification and transport measurement?
I don't think so. I mean, I don't think that the problem of heterogeneity and temporal variability is unique to the vertebrate system. I mean, you're going to be challenged with that regardless of what monitoring infrastructure you use. So if you use conventional wells, you still have that same challenge. And the only way to overcome that challenge is with adequate sampling density, both in space and time. So again, I think it sort of depends on the site-specific characteristics and the needs, but I don't see that challenge is inherent to this technology. It's going to be universal for whatever monitoring technology you use.
Wonderful. And this next question is from Jacobs. Have you conducted any tests to verify the integrity of the grout seals?
Yeah, that's a good question. We did play during some of the slug testing and pneumatic slug, both the falling head and pneumatic slug testing. We did have transducers in adjacent screens and did not see any hydraulic influence whatsoever. So, and then of course we don't have, we've got a lot of chemical variability preserved, so we don't think we've got failure of the seals at all. And also I should point out, we believe that you've got full formation collapse as well. So I don't think anywhere do you have open annulus. I do think there's places where the grout seal may be more developed than others, but in all cases you've got that annulus filled with native materials that have collapsed on the well construction. And we did some, I want to say 3 or 4 tests where we looked at response data and didn't see hydraulic effects.
Great. And then this next question from WSP, do you have any information on the long-term reliability of this monitoring system?
Yeah, again, I would defer to the folks at NREX. I mean, they've been putting these in the ground for more than 10 years. For the most part, they've been focused on hydrocarbon cleanup, like retail UST sites where you have a lot of access challenges, a lot of surface and footprint to go underneath. But they've got systems that are well over a decade, and they can kind of relay, any sort of long-term performance data from that. but I don't have any personal experience on it. But I see no reason why there's nothing in these materials that would lead me to think that there's any sort of unique life cycle to these. These should last decades easily.
Thank you. And the last question for you is just a request to please recap what kind of geological information is needed to support the design of the system.
Yeah, that's really important. I would say, as I pointed out, you're basically you have no ability to control the well design in the field. So you really have to know ahead of time exactly how you're going to install it. And so I would say in most settings, you're going to want, pretty high resolution information on both permeability so that you know your target lithologies. And in some settings, those target sand zones might only be a couple of feet thick. So you want good elevation data that, can be developed through HPT or CPT methods or other methods. And then again, that spatial variability and concentration, that also is important. And, what we're learning more and more is that there's more variability than we expected. And as I mentioned from our results, we had screens pretty close together. And even there, most of the mass discharge was being driven by 2 screens. So I would say it's really worth a pretty aggressive campaign ahead of time along the planned transect path. That includes high resolution geologic information and quite a bit of grab sampling. For PFAS, or of course this technology is contaminant independent. It can be used for any contaminant type.
Great. Well, thank you, Craig, for a great presentation and a very engaging Q&A. We're going to wrap up with one question for Rainer before the end of the webinar. So, Rainer, this is a question from the Department of Defense. You talked a lot about the benefits to DoD from using both equilibrium and integrative samplers. Are there any uncertainties associated with the use of these samplers?
Yes, that's a good question. So it's funny, as we develop these tools, we often have to justify how precise and accurate they are. But typically, we like to flip this around. And yes, of course, you can take a grab sample and the quality control is going to be much better on that particular sample. But of course, that sample reflects that point in time only. So I think from what I've worked on with passives, we typically say if we're within a factor of 2 of alternative means of measuring concentration, we're doing well. So I would say the same. They're not, because there's some more math involved, they're not going to be as precise as a measurement in space and time, but I think the information might be equally, if not more valuable, actually, to inform decisions.
Well, this is a fantastic response to the question, so thank you for that. We're going to go ahead and wrap up. Before we conclude, though, I'd like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future, as Andrea mentioned. And a reminder that the next webinar is an environmental restoration webinar. It's in 2 weeks on Thursday, September 21. This webinar will focus on DoD-funded research efforts on transitioning from active to passive remedies at DoD sites. The webinar will feature 2 speakers, Dr. Dave Adamson from GSI and Dr. Paul Tretniak from Oregon Health and Science University. Registration is open, so please visit the SERDP and ESTCP webinar webpage to register for this and other webinars through the end of this year. We would appreciate it at this time if you can please take a moment to complete the survey that will pop up on your screen. This concludes today's webcast. Thank you for joining us.